All right, hi everyone. Welcome back to our monthly spatial biology related meeting. So today we have Dr. Rong Fan with us. So Dr. Rong did his PhD in UCBerkley, and then he spent some time in CalTech doing his postdoctoral work. And now he's currently an associate professor in the Department of Biomedical Engineering at Yale. And his lab really works on developing a number of different single cell technologies to study cellular heterogeneity, intercellular signaling networks within in both human health and disease. And they have developed high throughput technologies for co-analysis of single cell epigenomics, transcriptomics, and proteomic profiles. He has co-founded a number of companies, and he's also a recipient of numerous awards, including NCI Howard Temin Career Transition Award, Career Award from NSF, the Packard Fellowship for Science and Engineering, and was recently elected as a fellow of the National Academy of Inventors. So without further ado, Dr. Rong, we are very happy to have you here, and I look forward to your presentation.
Yeah, thank you, Ajay. It's such a great pleasure being here, and thanks so much for the invitation. Let me go ahead and share my screen first. All right, can you see my screen?
Yes, we can.
But not full screen. All right, okay. Well, all right. I think my internet is a little bit slow. If you cannot hear me well, I'm gonna kind of turn off my video. I'm gonna go ahead and get started right now. So this is my disclaimer. I would like to start my talk with sort of the story I learned many years ago when I switched my career from physical science to biomedical research back to 2006. I graduated from UCBerkley and moved to the CalTech Jim Heath lab. And I was supposed to work on cancer biology or cancer technology, but I literally knew nothing about cancer. So I got a Doug Hanahan's the Bible review article, okay, the hallmarks of cancer. And I found I really cannot understand that much at all. And there's another one I read, I so much enjoy. And also I used this diagram over and over the past 15 years in many of my grant submissions. This is a review article from Professor Joanna Joyce. She was at MSK, and now she's Professor at University of Lausanne. And so in this article, basically I learned, okay, maybe you guys feel surprised, but this first time I learned the tumor is not a bag of tumor cells, actually many different cells there. And so you really needed tools to dissect that complexity or heterogeneity.
Yes, I met your children.
But there's another challenge we were not able to address back to that time. Even right now, still challenging is you need to have single cell resolution, but you need to understand how they organize in a tissue context in a spatial resolved manner. So my laboratory over the first kind of a couple of years at Yale was sort of the continuation of the focus I initiated at CalTech to look at the cell-cell interaction and the cell single-cell secretome, because I thought that's the sort of the language the cells use to communicate. Just like how we communicate in English, the cells don't speak English; they speak something called secreted proteins, cytokines, those I can put individual cells in the nanoliter microchamber, and by each chamber, I have a bunch of antibody stripes running perpendicular to the microchannel direction. And then I can have a whole set of antibody pattern within each chamber to do highly multiplex protein secretion profiling. So this technology kind of gradually evolved from this beautiful device I fabricated at CalTech, but when I moved to Yale, I still like this device. I decided to take this to my collaborators at Yale, but they have no idea how to use it. And I realized that's an engineering challenge. Since I'm in the Department of Biomedical Engineering, I think I should take on that challenge to really make the device much higher throughput, higher plex, and also much scalable and manufacturable as well. So over the past couple of years, actually first couple of years I was at Yale, we kind of demonstrate the technology sort of improvement over the prototype, but also the application in different biological questions, including very fundamental cell-cell interactions that, for example, right here, but also the protein secretion so from cancer cells and how the cancer secretome can basically alter the cancer microenvironment or tumor microenvironment. Very few people studied that, but that turns out to be quite exciting. But now this has been basically commercialized and widely used across the country and around the world to characterize immune cell function, and in particular in the cancer immuno-oncology space. But that's what I mean. So one aspect of the challenges I learned in Joanna Joy's review articles of you need a single cell resolution tools, but I just use my own example, but the major breakthrough in the field is the single cell sequencing technology, right? That allows you to look at the genome-wide gene expression or genome-wide epigenetic or genome-wide genomic features with single cell resolution. But another aspect is you don't really have spatial organization information since you dissociate the tissue completely. So this is how 10x Genomics describe the data structure from bulk sequencing to single cell and to the spatially resolved sequencing. So you probably know I love travel since I'm still in Cambridge, UK right now. So I tend to use this analogy. So this is the bulk sequencing data. You do know, okay, when you look at the shape of this map, this is USA. Single cell sequencing gives you a lot more information about what you have in this country. Okay, different states, different cities, New York City in New York, New Haven in Connecticut. But you're not able to drive around to enjoy your trip by having just this one and this one. What you really need is the full map, and with all the major cities connected and the freeway to tell you where to drive around. So it turns out biology works in the same way. Can we build the tissue map that looks like this full map of the United States? And so over the past couple of years, I think different technologies emerged to address this challenge, and basically creating this kind of burgeoning field. The next categories of the tools, broadly speaking, I think one is based on multiplex imaging, the other based on NGS sequencing, which is a little bit kind of latest technology innovation in the space. But even for imaging, about 12 years ago, even 10 years ago, if you are able to look at 5 different colors, you can get a very good quality image. People are very jealous. Okay, you are really an expert in this multiplex imaging. But nowadays, this has been kind of pushed to the entire genome or transcriptome scale. Two examples are here, the SIGFISH and MERFISH. You can do sequential detection of the barcodes, and then you combine the different barcodes to detect the different genes. That this combinatorial barcoding allows you to kind of map out entire transcriptome, but that's still very, very tricky, very, very challenging experiment. So what I would like to talk a little bit more in my presentation today is the high-throughput sequencing-based spatial omics. The landmark paper came out 2016 from Professor Joachim Lamberg's lab in Stockholm in Sweden. And so basically what he did is to use the DNA microarray spots to capture messenger RNAs released from the tissue section. You place them on top of the DNA microarray glass slide. And the transcript's going to be captured in the vicinity of those microarray spots, and each microarray spot has unique DNA oligo to determine the spatial location that basically serves as the spatial address code. When you do cDNA synthesis on the glass slide, you're going to incorporate those cDNA. You're going to incorporate those DNA oligos on the spatial address codes into your cDNA, and then you collect all the cDNA and amplify by PCR and make the sequencing library and perform parallel sequencing. You are able to read the spatial address codes and also read the expression of different genes. When you combine both, you get a spatial resolved gene expression profile. But back to that time, I think the spot size was like 100 micron. And when I talked to my colleagues at Yale, they were not that excited about this technology back to that time, but that's not the end of the story. The technology continues to evolve, and over the past couple of years, including the commercialized 10x Visium and the SLISIC and the HDST from the friends in Boston. And last year, and some of them actually just published a month ago. And the three new technologies allow you to make the spot size to reach the spot size even smaller than 1 micron. You are getting subcellular spatial resolved transcriptome sequencing. So this is what I kind of already described. So how the technology works, using the barcode solid phase RNA capture for spatial transcriptome profiling. This is what Professor Joachim Lamberg described. This terminology kind of coined by Joachim Lamberg. Even though now the spot size is getting smaller and smaller, and how you're making the spots, and it's a very, very different approach, but the fundamental principle remains the same. And so what we came up with a couple of years ago was kind of totally opposite. Instead of taking messenger RNAs out of the tissue and capture on a solid substrate, we send the DNA barcodes into the tissue to barcode whatever information you want to barcode directly in the tissue matrix. So that time, about 2 years ago, we demonstrated we can barcode an entire transcriptome and also a panel of proteins, kind of 22 proteins, but that's not the limit. And the first demonstration, we can do the spatial multi-omics and whole transcriptome and the proteins. And so recently we further demonstrate this is so versatile approach. You can basically barcode many different omic information, which I'm gonna talk to you in my presentation. So how come we came up with this idea? This is sort of related to what I told you in the very beginning. We developed this antibody barcode microarray to detect the single cell secretome. The same device, the same antibody microarray was used to detect a large panel of proteins from finger prick of blood. So the fundamental or foundational technology enable both of that. this microchannel guide delivering a pattern of DNA or proteins to perform spatially encoded highly multiplex biological assays. So now you think about if the tissue is in the substrate, do microchannel guided delivery or pattern of DNA molecules can directly deliver those DNA oligos or spatial address codes into the tissue in a highly multiplex and spatially resolved manner. So this is a device we developed to perform that spatial barcoding in tissue. And so honestly, this is a very simple device. I have been working on microfluidics over 15 years. This is really the simplest microfluidic device I ever worked with. You don't need a complicated fluid handling system as long as you can pipette reagents into those inlet holes and a global vacuum and a bag to put reagents into the channels over the surface of the tissue. You are able to perform this assay. So how the workflow, how the assay works, I think if you have the tissue on a glass slide, that's totally fine. You don't have to bring your device to a cryo-sectioning machine. Okay, you don't have to kind of precisely kind of land your tissue section on the vision kind of little square. You can put it anywhere as long as that's close to the center of the glass slide. It's good enough. And then you put the first microfluid channel device on the tissue section to introduce the first set of DNA barcodes, A1 to A50. So now what we are doing is A1 to A100. And those DNA barcode sequence have the oligo DT that bind to the poly tail of the cDNA, but you can also play in a trick that it's very much like the CySig, using the DNA antibody conjugates to detect the proteins. Those DNA oligos tethered to the antibody have no poly tail, so you can use basically the same chemistry to barcode to detect those proteins. But that's only one direction, right? So you're removing the DNA, you remove the microfluid device and put another one on top of the same tissue section with the channels perpendicular to the first flow direction to introduce the second set of DNA barcodes, B1 to B50 or B1 to B100. And then you ligate them together and the intersections, creating this 2D grid of tissue pixels. And after you remove the second PDMS device, your tissue is actually still there. You can image, you can actually see those little tissue pixels. And each tissue pixel has unique combination of A and a B, which gives you this spatial address code. Finally, you just collect all the cDNA and the PCR amplify and then making a library and put in the Illumina sequencer for pairing and sequencing. And then you can computationally reconstruct the spatial gene expression or spatial protein expression. So it turns out this in-tissue barcoding is pretty versatile. Last year, I think 2 papers also adopt this in-tissue barcoding, even though they are not using microfluidics, but the fundamental principle is the same. So you send the DNA barcodes into the tissue to in this case, to barcode the transcripts, and but in the phase change lab, they also demonstrate can barcode the genomic DNA as well. And okay, so that paper has been published. I decided not to talk about any data from that paper, from that Cell paper. I just show you what I have gone further since then in either the multi-omics protein and the transcriptome, but also beyond the whole transcript and the panel of proteins. So recently we mapped out the human hippocampus tissue. I think a very beautiful data, but I don't want to go into the details. This is look at this, you appreciate this sort of the clustering results then you project the different clusters back to the tissue. You see very nice spatial features that corresponds very well to the anatomic annotation of the different regions in the human hippocampus. So we did the same experiment, 10 different human brains. 6 of them are from the healthy donors, and 4 of them are from major depression disease patients. It's a very interesting difference we observe. I think stay tuned. We probably want to release the data in the next month or so, if you're interested in the neuroscience spatial omics. I think sort of a year ago, more than a year ago, I think the spatial transcriptome was selected as the method of the year by Nature Methods, and in the same issue, the Nature Methods highlight our work and saying, okay, multi-omics sequencing goes spatial now. So now the question is, we talk about multi-omics, proteins, and the transcriptome, but that time you only demonstrate 22 proteins. How many more you can do? There are fundamental limit there, or actually you can get 100 or even 1,000. I don't have an answer. I don't exactly know, but I would like to try whether or not we can get at least 200. So we released this bioRxiv paper a couple of months ago to show the hundreds, up to 300 proteins we mapped out using this DBasic approach in conjunction with the whole transcriptome. And the data look very good from a variety of different tissue types. I think in this case, different mouse tissues, but also we demonstrate that in different human tissues as well. So actually around the same time, I think Dan Landau's lab at the New York Genome Center also kind of demonstrate this multi-omics transcriptome and a panel of proteins using conventional text vision platform. If you're interested, you can check out this bioRxiv paper as well. And so in our spatial SciSig, we demonstrate the 300 protein spatial mapping and the whole transcriptome on the human tonsil tissues. In this case, you can see the B cell follicles and from the tissue image, but from the sequencing data, you get 300 proteins and a whole transcriptome. Because you have a large number of proteins, we decided to perform unsupervised clustering of just the protein data only. So we found we can get a very nice clusters, about 7 different clusters, and we project them to the tissue image. We say, wow, the really kind of corresponds so well to the different tissue regions. This B cell follicles, and this kind of extrafollicular T cell zones. You see the epithelial crypt region and some vasculature there. And so from the whole transcriptome, you see very similar spatial features, but somehow I think the protein data give you better spatial resolution or spatial distinction of different clusters. However, I don't want to say the proteins work better than the messenger RNAs for the sort of the different tissue feature identification. Even though we use only 10% of the sequencing length, we detect only 300 proteins. So in this case, we have much deeper sequencing depth per protein as compared to per gene in this data. So now you can look at all different proteins one by one, and 300 proteins across this entire tissue region. And so it's, I think it takes forever, okay, to show you all of them. I just show you a couple of kind of marker genes, for example, the B cell CD19, and also the functional B cell genes, different immunoglobulins, IgM, IgG, and IgD. IgD produced by naive B cells, you do see those cells are kind of getting into the tonsil tissue from the vasculature. And the IgM producing B cells are in the B cell follicles, and a little bit more mature B cells that can produce IgG already migrate out of the follicles and migrate out to the most regions of the tonsil. And also you can look at the T cell zones. I think the CD3 T cell marker, you can also look at a specific subset of T cells like helper T cells. You do see some relatively sparse T cells in the follicles, and very likely those are kind of follicular helper T cells. And many other things, I think not just the T cell, B cells, but also the epithelial, the stromal cells, you can see where they are based on all those different protein markers. So another interesting example is the human skin. So in this case, we use exactly the same panel, 300 proteins. And this panel is based on the BioLegend total seq panel. So all the proteins all the DNA antibody conjugates they have, we told them, okay, can you make a cocktail just to give to us? And so in this case, for all the genes, all the transcripts, or all the proteins, we perform unsupervised clustering independently and project those clusters to the tissue. So you can see how well the different spatial clusters kind of match each other between the two different data sets, the transcripts versus the proteins. We can resolve all different major regions. So even though for the transcripts, I think in this in the kind of low cell density region, you don't have a lot of genes, but you still get a similar kind of spatial features or spatial patterns between these two different data sets. Actually, we saw a lot of interesting things like local T cell, peripheral T cell activation early on once you have the Moderna vaccine injected to the underneath the skin, but I'm not going to talk about that details. Okay, maybe next second half of my talk, I would like to share with you, so beyond the spatial transcriptome and the panel proteins, what else we can do. In my lab, we have been very interested in epigenetics. There are many different epigenetic regulations you can look at, and histone modifications and DNA methylation and the 3D organization of the chromatin structure and so on and so forth. We decided to look at this two first. Okay, one is the chromatin accessibility. And the second is histone-specific histone modifications or different histone marks. And so the first is global accessibility of the histone, the histone modification, the histone, the chromatin accessibility. And so in this case, I think Will Greenleaf at Stanford, I think, and also Jason Broad, and they developed the ataxic, and then later they further developed the single cell ataxic. We wonder whether or not we can do spatial resolve the ataxic direct on the tissue surface and pixel by pixel without tissue dissociation, and then we can resolve the epigenetic state directly. We can visualize the epigenetic states directly in the tissue. So it turns out the chemistry is even simpler, and it's a pretty straightforward ATAC chemistry. Okay, use TN5 transposase pre-assembled with the DNA oligos to train the tissue section in this case. I think in the past, turns out no one tried to use the TN5 transposase to process, to train the tissue section. They always, okay, dissociate into single cell or single nuclei and do this transposition chemistry. But we found actually that worked out very well. Okay, don't have to dissociate the tissue, just make a very thin section and put the reagent on top, and you're going to see the TM5 transposase cutting the open chromatin sites, and then insert the DNA oligos to the genomic DNA and open chromatin region. So actually, if you just want to get ataxic data, you can dissociate the tissue and perform sequencing. But in our case, we perform 2 more steps to add a barcodes A and a B. For each of them, we use the temporal ligation to add the barcodes A to the DNA oligos. We kind of insert it into the genomic DNA and then ligate barcode B to barcode A to create this kind of 2D grade of the tissue pixels. So now solving every experiment, we have thousands of ataxic experiments. Each corresponds to 1 tiny tissue pixel, so we can map down on a entire tissue region pixel by pixel. So from the data we collected from a mouse embryo, and in this case, we look at a specific region of the liver, embryonic liver. We have a lot of different pixels you can aggregate to make the pseudo bulk ataxic data. And looking at data trace, that looks very much like the bulk encoded reference data. But if you take a closer look, actually we have even better signal to noise ratio compared to the encoded reference data. So using all the tissue pixels, their ATAC data from every single pixel, we can perform unsupervised clustering of all the pixels. So this is what you see, and you put them back to the tissue to look at where they are. You do see very nice concordance between different clusters and the different tissue regions. For example, this is in the spine, and this is the neural tube, and you see the different internal organs, and very interesting, many different tissue types already developed in the embryonic mouse brain. You can look at individual genes, but now the gene expression, what you see here is the transcription starting site enrichment. So you can look at them one by one, and this is a gene very specific to the hematopoiesis in the mouse embryo that actually occurs in the liver early on in the embryo. So this gene corresponds to the spine, and actually don't really see that from the encode reference data because no one actually dissected the very, very thin spine to perform bulk ataxic encode in the encode project. So you see a lot of interesting features in the brain, in the CNS development. I'm not gonna go through the details, but you'll see how nicely you can visualize individual genes in terms of the epigenetic state. So again, we use the encode reference data to benchmark what we can get from our spatial ataxic, which is right here. This encode reference data, we combine them to perform unsupervised clustering. And so you see pretty good concordance, but overall you'll see a lot more granular information because we got the data pixel by pixel, but in the encode reference data, they physically dissect out the different parts of the tissue. So that's kind of bulk now single cell, and now now the spatial resolve. But you still see pretty a good concordance, sort of which cluster corresponds to which. For example, the limb versus limb here, liver versus liver in this cluster. And so what is much more powerful is to combine the single cell sequencing data. In this case, we combine the single cell transcriptome sequencing data. We also perform single cell ataxic data integration, but just to show you the integration of the spatial ATAC and a single cell transcriptome. So people have done very nice sort of cell type annotation from the single cell transcriptome sequencing of the mouse embryo at this stage. And then we can take advantage of what people have done. This is from Jay Sendra's lab. They have annotated all different cell lineages, different cell types. So now when we put them together, on the spatial pixels are those black dots. You just look for the overlap between the black dots and the different clusters identified or different cell types identified by single-cell sequencing. And then you can, for example, right here, if that's the cluster identified by the single-cell sequencing or the specific cell type identified by single-cell sequencing, okay, those black dots must have the dominant cell type determined by that specific cell lineage. So in this case, you can use very simple label transfer to identify major cell types in every single pixel. So I just show you a couple of those, and then you can go back to check where they are. And for example, the definitive urethral lineage, and they are enriched in liver. Okay, that's exactly what we expected for the mouse embryo at this stage. You see epithelial cells lining around those kind of the limb, and it's also part of the sort of the skin right here, and etc. neurons and other different neuronal cell types like post-mitotic premature neurons. And then this oligodendrocyte progenitors are kind of lining around the ventricle, and that's sort of exactly what people kind of speculated, but in this case we can directly visualize. But keep in mind what you are seeing here is not a gene expression, but actually the epigenetic. So whether or not the epigenetic state controls the development of that particular cell type or cell lineage in this tissue region. So we did another experiment with the postnatal mouse brain, okay, not mouse embryo. So in this case, we look at several different regions, but I just show you one of those right here. And so this interesting region covers this sort of the area we mapped out cover multiple interesting regions like a kind of inner part of the cortex and a corpus callosum and a striatum, and right here this kind of the lateral ventricle right in between. And so from on the pixels unsupervised the clustering, we can identify the major clusters, and then you go back to see where those clusters are in the tissue. You do see very nice concordance between the different regions people have prior knowledge about, and then you can now basically visualize whether or not indeed those epigenetic state, in this case open chromatin state, determines the different tissue types. You can visualize individual genes as well, and even though in this case very, very thin layer of the cells lining this kind of neuroepithelial layer, you can visualize using this spatial ataxic that at a resolution of 20 micron. And it may not be necessary to go further lower or further higher resolution or further smaller, because 20 micron, we do see a lot of single nucleus pixels. That means you do have single cell or single nucleus spatial ataxy data directly in the tissue in situ. All right, so again, you can integrate the single cell sequencing data from exactly the same mouse embryo, the stage of postnatal mouse brain, either spatial attacks, either single cell sequencing data with the spatial attack or single cell ataxy data and the spatial ataxy data integration. I just show you one of them. That's just a single cell sequencing data to identify all different cell lineages at this stage of the postnatal mouse brain. And then you look for overlap, okay, between different spatial clusters right here and here, and you know where those different cell lineages are located in this region of the tissue. So those are different cell lineages, and so you can see where they are in the tissue. So the last example is, so what I'm very interested in, I'm always interested in is lymph tissue, tonsil, lymph nodes, bone marrow. So again, there's a human tonsil. We did a spatial ataxic. We use relatively low resolution clustering to identify the major tissue regions or major clusters. So 3 major clusters here correspond to the B cell follicles, extrafollicular region, and also the epithelial crisp. And so you can look at individual genes. In this case, you are not limited to the surface markers or sort of the proteins you are able to detect using DNA antibody conjugates. Basically, there's a global unbiased genome-wide spatial mapping, but you are mapping the epigenetic state. You are not mapping the gene expression. So from those specific marker genes, for example, CD10, this is GC, germinal-center B cell marker, and it's strongly enriched in the B cell follicles. That means, okay, indeed, this specific gene is determined by the epigenetic chromatin accessibility. And now you can look at not just the surface markers, but also for example, PAX5, the master transcription factor associated with germinal center B cells that's strongly enriched in here. And you can look at some T cell markers, CD3, CD, and the different T cell differentiation markers, CCR7, and this is CCR5, and also the BCL6, which is the master transcription factor for follicular B cells, but also follicular helper T cells. It's exclusively, I think, accessible in a germinal center or in B cell follicles. I think an interesting finding from our data is the FOXP3. So if you do immunostaining, which we did, and or you do FISH, to look at FOXP3 positive T cells, you don't see a lot. So, but from the epigenetic state, you see a lot of epigenetic prime or epigenetic rated FOXP3 T cells, but there are not really Treg. They are not really regulatory T cells, but since they have the chromatin open to quickly express the FOXP3 if needed, so they are ready to turn themselves into Tregs. So that's my understanding. I think this is a very smart in this tissue, in this type of cells. So they always have those epigenetically ready T cells in the extrafollicular region. If you see kind of hyperinflammatory response in the tissue, and those cells, they don't have to go through lengthy phenotypic transformation. They are ready to turn themselves into regulatory T cells to counteract the hyperinflammatory response in the tissue, in the tonsil. That's my understanding. I really think it's super fascinating. So you'll see different messages from the epigenetic spatial mapping versus gene expression or transcriptome spatial mapping. So you can look at many other cell types, not limited to the lymphocytes, B cells, T cells. Also, you can integrate the single cell transcriptome sequencing data. I'm not showing you here, but that's exactly what we did to do the data integration, label transfer, and then we can identify different cell types like naive B cells, GC B cells, macrophage, and then you'll see where they are. And you can also perform unsupervised, you can also perform cell differentiation analysis to look at the differentiation all the way from naive B cells to germinal center B cells. You see the differentiation schedule time trajectory. But what is cool in this case, you can see where they are. Okay, you see really the differentiation trajectory spatial, okay, from the extrafollicular to the follicular region, you see the naive B cells all the way to germinal center B cells. Okay, I spent a little bit more time on spatial ataxic because that has not been published. I would like to share some unpublished data. So we're able to do specific chromatin modification spatial mapping as well, and this work has been published earlier this year. So then I think I can go a little bit faster, and if you're interested, you can check out this paper. So in this case, we did different histone marks, H3K27 trimethylation, K4 trimethylation, and K27 acetylation from kind of several tissue sections. And so in this case, you even without multiplex, so multiplex chipstick or multiplex cut and tag from the several sections, you can sort of see the multivalency of different histone modifications. So for example, here, I think this histone mark doesn't really resolve the different tissues between this region and here, but this histone mark that does, and this histone mark even cannot resolve those. So when you combine them, you really see how different histone marks controls different tissue formation. So this is sort of much more insights in terms of the epigenetic regulation mechanisms. The data quality is extremely good, and compared to single cell, it's completely comparable to the best single cell cut and tag data. It's even slightly better. And so we compare our data to the ENCODE reference data from the same organs at a different the same mouse embryo, the same stage of the mouse embryo. So again, you can see the clusters associated with liver from two different data sets. For heart, they do show pretty good concordance between the reference data and our spatial cut and tag. So you'll have the single cell sequencing data, in this case, single cell transcriptome. You can do data integration to look at different cell types. Basically, in the single cell sequencing data allows you to do the cell type annotation. Again, this was done by Jay Sandru's lab. So now you can look at all different cell types and see where they are if you see them, okay, by kind of overlaying these two UMaps, you'll find the overlapping region between the different clusters identified by single cell sequencing, and also from our spatial clusters. That's how you can sort of use the label transfer to identify different cell types. But again, I would like to emphasize what you are seeing is not just a gene expression and different cell types determined by either transcriptional proteins. What you see is whether or not this specific histone modifications controls the development of that particular cell type. All right. And so similarly, you can perform the differentiation or shorter time analysis. So what you see here is, okay, all the way from the ventricle, sort of the layers lining around the ventricle all the way into the brain. You can see the radioglia to the excitatory neurons, and also in this case, what you know is not just in the different cell types from the progenitor to the more mature neurons where they are, but actually what you know is, hey, this histone modification is relevant. This histone modification is associated with this cell differentiation process, or very likely controls this cell differentiation process. And so I'm probably going to skip those because this has been published. So again, we mapped out the mouse brain. So postnatal mouse brain, I believe it's the P25, and from this one single histone modification spatial mapping unsupervised cluster. You can see very distinct spatial clusters, spatial features from all different clusters here. So I'm gonna just skip those just to give you sort of the final punch. So people constantly talk to me, so you are able to map down the spatial transcriptome and the spatial epigenome. Can you combine? I would say it's not difficult at all to combine. We just want to do this step by step. But I would like to show you some exciting data from the spatially resolved epigenome transcriptome code sequencing. So this is kind of the workflow. We're able to do both ATAC and the RNA code mapping and a histone modification and the transcriptome code mapping. So this is the data from one of the most interesting tissue samples. This is a human hippocampus. So we basically did just a one experiment to map out both. This ATAC, this is from the transcriptome, the unsupervised clustering, and you see where the different clusters are. You basically appreciate how kind of strong concordance between these two different modalities to identify different tissue types, different cell types in different tissue regions. And this is the initial staining from the adjacent tissue section. So now you can see the different layers of the hippocampus, human hippocampus, and different tissue regions, and we can resolve using either one of those two modalities. But again, we can also use the single cell sequencing data to perform the cell type identification, label transfer, and to visualize where the different cells are. That's very similar to what we described in sort of spatial CalTech and spatial ataxic. But now you have both. All right, so we're trying to do this sort of peak to gene linkage analysis. We identify more than like 15,000 different peak to gene linkages. That's pretty complicated bioinformatics, but to make it simple, so if you have both, can you just go ahead and visualize those individual genes at a different levels, like epigenetic and a transcriptional? Okay. Let's just show you a couple of examples. In most of the cases, actually, we do see pretty good concordance, but really picking the ones that are not perfect concordance. And for example, this one, I think a PROX1, and this has been studied over and over in the human brain. And we know this is canonic marker for granular cell layer. And for granular cells. And however, when you look at the epigenetic open chromatin state, you don't really see PROX1 strongly enriched in the granular layer. But you do see the expression of the PROX1 when you look at the RNA data. So now we kind of hypothesize maybe this canonic marker expressed and the cells are post-mitotic. I think most of them are neurons, very mature neurons. You don't have to have the chromatin open because, okay, those cells are there, they are PROX1 positive. So in this case, you may further speculate those cells should show extremely low turnover rate for this particular transcript PROX1. And then we decided to look at the RNA velocity to look at sort of the RNA turnover speed or turnover rate. And this is RNA velocity analysis. Okay, this is a granular cell layer. This is sort of the CA. This looks exactly like what we expected. And from the flow of basically different cell types to more mature neurons. But now we revisit PROX1, we see, indeed, okay, PROX1 enriched right here gives you extremely high expression level. But when you look at the turnover and the velocity, it's relatively low, okay, compared to other regions. So this really confirms, so the different layers of the omics, you see different things, okay? Don't anticipate or don't ask, okay, which one works better to identify different cell types. Okay, epigenetic versus transcription, you really see different things. Okay, well, Greenleaf always use the peanut butter jelly analogy. Okay, you really need both to make it a perfect sandwich. All right, so basically that is, I hope this is a fascinating so preliminary finding. I share with you, you appreciate this spatial multi-omics, in particular spatial epigenome and transcriptome co-profiling that can help you better understand the mechanism directly in the tissue context. So over the past years, we have seen this kind of explosion of the single cell and now the spatial technologies. So the leaders in the field, not just in academia, but also in industry, they really believe we're right at the beginning of another wave of biology revolution or biomedical research revolution, largely because we have this kind of spatial or spatial single cell technologies that are becoming available. So a lot of new opportunities. I don't think I have time to further discuss with you, but I just show you what I like. This is a few examples. I would like to use this slide to kind of conclude, to summarize what I talk about in the NGS-based high-throughput spatial omics sequencing space. So what my laboratory developed is kind of completely different, unique approach to do spatial omics analysis by sequencing. And turns out it's extremely versatile. You can look at the transcriptome proteins and many, many other things. And even you combine them to do multi-omics, epigenome, transcriptome, and even a large panel of proteins. That's totally durable. It's not much, it's not difficult at all. All right, so yeah, I would like to thank the people in my lab over the past years who contribute to the technology development, but also my wonderful collaborators who help us to apply the technology to different biological questions, including my collaborators at Yale, but many of them outside Yale. For example, Gonzalo from Karolinska Institute, I never anticipated during the pandemic. I think in the collaboration across the Atlantic Ocean can work out that well, but turns out yes. And I would like to thank the funding agencies for the support, and thank you very much for your attention. I hope I have a little bit of time to answer your questions.
Yeah, thank you so much, Rong, for giving this exciting talk. That was really, really impressive what you can do. And yeah, you're right that I also had the question of like, can you actually combine ATAC-seq or spatial ATAC-seq with spatial transcriptomics? And it's really exciting that this is possible. Moving forward, of course, the next question is, can you actually combine this with proteomics as well? Is that something you're working on or you think could be possible at all?
Yeah, it's totally possible, and the two ways to do, I think. One is kind of combine spatial sci-seq and this spatial epigenome transcriptome co-sequencing. I don't see any difficulty to do that, but just right now we are working on the brain samples, and most of those antibodies are really for immune cells or for lymphoid tissues. Probably we're gonna do either tonsil or lymph node for that experiment rather than mouse brain, human brain. But maybe that might not be true. Okay, you can still see a lot of microglia and other immune cells in brain. Yeah, we'll give it a try and let you know. The approach number two is actually can do multiplex imaging. For example, either like a CySIF or Codex, Well, particularly interesting, can we do both on the same tissue section? We're getting there, but if not, we can at least do this protein from adjacent tissue sections. But I really think we can do that from the same tissue section, since we do in tissue barcoding, and if you can retrieve the cDNA and get the barcode DNA out without damaging the proteins, then you can still have those the tissue section with no proteins there to do multiplex immunostaining, such as CySF.
Yeah, cool. That's really exciting. Thank you so much. We have a lot of questions also in the chat. Whoever wants to actually unmute yourself and ask their question, please feel free. Just raise your hand. Otherwise, I may just start reading some questions. So,
Hi,
Jasmine.
yeah,
Yes,
hi, it's Jasmine. Hi, Fanyan. I've looked at lots of and I looked at a lot of your stuff. So I saw you on another talk, but I had a really specific question. So I apologize to the rest of the audience. How big is the capture area? Because that's what we're all talking about, right? So
yeah, you mean the tissue mapping area,
yeah.
right? Yeah, it varies because in our case, if we use 100 by 100 barcodes. Yeah, if that's 10 micron, it depends on the pixel size, right? So we always have 100 by 100. It depends on the pixel size. If that's 50 micron pixel, you are getting close to 1 centimeter. So that's what we are doing for some human brain samples. If you are only interested in neurons, okay, now those tiny cells there, 50 micron gives you pretty interesting data, and in that case you can map out 1 centimeter by 1 centimeter. But if you really need a 10 micron, even smaller pixel size, you probably can do only 2 millimeter. However, we have other approaches to increase the mapping area. For example, we can make microfluid channels kind of back and forth in the serpentine design, which I did during my postdoc for this antibody barcode device. So we do have that device in the lab, and that's totally durable. But end of the day, you have to spend 10 times more cash on sequencing. So just you'll probably need to find the right balance.
Yeah. It was great. Thank you.
All right, thank you. I think we also, Teresa, I wanted to ask if you can put your Outlook slide up while you're answering questions, but if not, that's fine. We can put also the recording online, so you can check that too, but it's fully up to you, Rong. So we have 2 questions from Joshua Lexina. If he wants to unmute, if you want to unmute yourself, feel free.
Yeah, I can read as well.
Yeah,
Okay.
this was a great talk.
Okay.
Yes, thanks so much. Yeah, my first question was about the effects of different tissue preservation techniques. And I saw on your last slide, you alluded to this high sensitivity FFPE DBIT-seq, which I'm interested to read more about. But what have you seen in terms of, fresh frozen versus fixed frozen versus the FFPE for the different outputs of this platform?
Okay, that's somehow, I think we have done a lot of spatial tissue sequencing, but we haven't processed any single tissue samples by ourselves. We always rely on collaborators. So in that case, that means probably our platform is quite compatible with many different tissue preps, since different labs, they have different preference. But so far, I think fresh frozen works well, and I don't know the difference between the flash frozen, fresh frozen. But FFPE, we have pretty good data from FFPE in some cases, but some cases not. So that's why, due to the limit of time, I decided not to talk that much, because in this presentation, I think we still need to know what's the sort of tissue quality metric you can use. So it turns out the sample rate number is not a perfect indicator of the tissue quality for spatial sequencing and some low rate number samples, we got a pretty good data. Yeah, I still don't I still don't know. I don't have the perfect answer yet. But we do have some data, even from clinical tumor specimens. We got like a 10,000 UMIs per 10 micron pixel, which is very, very good.
That's great. And it's related to my second question, which is how much do you have to customize the conditions for the microfluidics when you're working with for different tissue types, particularly ones that have a lot of connective tissue, like you had the skin data, but also tissues like the heart?
We have amazing data from heart. I don't have time to talk about. Yeah, I think how to process the tissue, how to make the sections, I don't know. I think always our collaborators, but as long as you can put the tissue on a standard glass slide and as long as kind of nice clean glass slide and you don't see delamination, that should work. We do have a little bit of experience, kind of failure experience of a very collagenous tissue like a retina. We still cannot get it work because I think the back side of the retina, it's just a very thick layer of the collagen. Yeah, somehow in that case, we were not able to get a perfect flow of the DNA barcode solutions across that layer of collagen tissue. So.
All right, great, thank you.
Can I ask a question?
Yeah, sure.
So with your combined RNA and nucleus data, you argued that for the nucleus, you don't need deconvolution because most of the time you only have one nucleus. For RNA, also for slide 2, you always get like mixtures. Have you tried to inform your deconvolution using the nucleus data?
This is a great question, actually, but I want to clarify a little bit. I don't want to say we don't need deconvolution for the epigenome data, and even though we do see a lot of single nucleus pixels, but still not the majority, and probably only 20% of those pixels. So I would like to see how to do deconvolution in D. And I actually tried once, but that didn't work. Seems like, yeah, we're not computational biology lab. If anyone would like to develop that pipeline for us, that would be great. But I would say in D, so if we have the single nucleus data, if we can kind of image exactly what we have in every single pixel, right? And you have the protein data by imaging, and you have the transcriptome, and then you have that single nucleus there for ataxic. I really think all data combined gonna give you a much better kind of guided deconvolution rather than, okay, now just a random, okay, the single cell data set, you have the spatial transcriptome, you just do kind of de novo decomposition or deconvolution. I really think this kind of guided deconvolution should work a lot better. But yeah, I'm not expert. I don't know how to do, but I just put out there.
I think there was one question from Ajit about the FOXP3 signal.
I was just wondering, is there a possibility that it could be an artifact?
Yeah, we didn't stand exactly the same tissue section, but we stand adjacent sections. No, very, very low FOXP3 and a protein level. We didn't do fish. So I think unlike that's artifact, and
I think
yeah,
the reason why
and we see that pretty often. We also see that pretty often in the spatial ataxic data of human tonsil. Yeah.
Awesome. Thank you.
Mm-hmm.
Yeah, thank you so much. Again, I actually wanted to ask one final question. So for some some researchers wanting to use your technologies, what is the best way to collaborate with you? Or is it even possible to do, for example, the combined spatial proteomic and transcriptomic profiling in your own lab without any special equipment, or do you need a microfluidic system? What is the best way to use your technologies or work with you?
Yeah, come and learn. I know Peter Soger's lab that organized that SciSafe bootcamp, something like that. That's fantastic. Just somehow due to the pandemic, we're not able to do so. But now, yeah, we are pretty much kind of hosting visitors every week, and interesting. Most of them are from outside US, from European countries.
I was wondering if it would be easier too to partner with like Cold Spring Harbor or something, because they're doing so many single cell workshops and alluding to spatial. You would be the perfect.
Oh, that's right. They have the single cell course, right? Yeah, I'm happy. Yeah, happy to do. Yes.
That might be an easier way than overloading your one poor lab.
That's right. Yeah, that's terrific idea.
Great.
So when is next the single cell course?
They do it in the summer, so they just wrapped it up. So I think
okay,
perfect timing to try maybe plan it for next year, so that you're gonna have to do everything.
Yeah, yeah. Next year we can teach a lot more. Maybe spatial omic.
That would be great. Well, thank you so much again, Rong. That was spectacular. And thanks so much for staying on a little bit longer for answering our questions. And also thanks everyone who joined the meeting. And yeah, I don't know, Ajit, do you have a final note?
I just wanted to thank Rong once again. Thank you for accepting our invitation and coming over. Thank you so much.
Yeah.
Right.
Thank you, Ajit. Thank you, Mary.
Thank you, everyone. Bye-bye.